So we are discussing about the biotechnology. You have seen the branches of biotechnology and applications of biotechnology in that. So interference completed, and then remember monoclonal antibodies. Monoclonal antibodies. Monoclonal antibodies. First of all, you have to know what are antibodies. So antibodies are the protective proteins released by our immune system to fight against the antigens. So here you have to remember these terminologies: antigens and antibodies. Antigens and antibodies. So what are antigens? Antigens are the foreign particles which enter into our body, which causes harm. That is called as an antigen. Or otherwise, antigen is a foreign particle which triggers our immune system. Which triggers our immune system, which triggers the immune response. That is called as an antigen. Simply remember, antigens are the foreign particle which causes harm or which triggers our immune system. So they are called antigens. So these antigens may be of different types. So chemically they may be carbohydrates, they may be proteins, or they may be some other chemical substances. But what are antibodies? These antibodies are the protective proteins. Protective proteins released by our immune system to fight against this antigen. Antibodies are the protective proteins released by our immune system to fight against the antigens. They are called antibodies. Generally, in our body, the antibodies released are polyclonal in nature. Polyclonal. Generally, the antibodies released in our body or the antibodies released by our immune system, they are polyclonal. Polyclonal means, so actually we have WBC cells, white blood cells. So these are the protective cells. In these WBC cells, we have lymphocytes. Lymphocytes. So these lymphocytes, they releases the antibody. There are two types of lymphocytes. Just to recollect this one. B lymphocytes and T lymphocytes. And these B lymphocytes will releases the antibodies. Okay. There are different kinds of B lymphocytes. Each B lymphocyte releases a specific antibody. So like that, there are different kinds of B lymphocytes which releases different types of antibodies against two different antigens. Is it clear? So these antibodies which are released from different types of B lymphocytes, they are called as polyclonal antibodies. Are you getting? But here we want to produce these monoclonal antibodies. So simply, this is the brief introduction. In our body, the antibodies released are polyclonal in nature. Polyclonal in nature means different types of B lymphocytes will release us different types of antibodies, and these antibodies we call as a polyclonal. They are specific against a specific type of antigens. These are polyclonal in nature, just to remember that. But here we are discussing about this monoclonal antibodies. Monoclonal, mono means single. Clone means cluster or a group. Here, suppose say this is B lymphocyte. Say this is B lymphocyte. There are clones of B lymphocytes. There are groups of B lymphocytes, and these B lymphocytes will release a specific antibody against a specific antigen. So group of antibodies are released from a group of cells. All these cells are called as clones. Clone means what? All these cells have similar in their genetic nature, similar in their structure and function. These are called clones. So from this clone, a similar type of antibody is released against a specific antigen. And all these antibodies we call as monoclonal antibodies. Means, simply you remember, monoclonal means from a single cluster, from a single group of cells, the antibodies are released which are similar in their structure and function. And these type of antibodies we call as monoclonal antibodies. So we can raise an antibody against a specific pathogen, specific antigen. Suppose, say for example, hepatitis B is there. That hepatitis B is a disease caused by hepatitis B virus. So we can raise the antibodies against to this hepatitis B virus. Okay, all these antibodies are similar in the structure and function, which are functioning against hepatitis B. These are called as monoclonal antibodies. So simply blindly you remember, the antibodies released in our body are polyclonal in nature. Different B lymphocyte cells will release us different types of antibodies. Otherwise, suppose say this is an antibody, it is having polypeptide chains. Polypeptide chains means so a group of amino acids. So each polypeptide chain is released by an individual B lymphocyte. So that's why they are called polyclonal. But here it is not like that. That means a single group of B lymphocytes will release us similar type of antibodies. These antibodies are called as monoclonal antibodies. Otherwise, the homogeneous preparation of antibody molecules from a single B lymphocyte is called as monoclonal antibodies. Now, here, in example point of view, you have to remember the monoclonal antibodies are produced by hybridoma technology. Hybridoma technology. This technology you have to remember. Monoclonal antibodies are produced by hybridoma technology. Just to remember this terminology, hybridoma technology. Monoclonal antibodies are produced by hybridoma technology, or otherwise hybridoma technology is used to produce monoclonal antibodies. Then let us see what is hybridoma technology. Here, the B lymphocyte cell is there. Say this is B lymphocyte or B cell. B cell. And B cell is having antibody producing capacity. The B cells can release the antibodies. Okay. And another cell is it, that we call as myeloma cell. So this is another cell. This is called myeloma cell. So what is myeloma cell? Myeloma cell means it is a cancer cell. What is the literal meaning of cancer? Uncontrolled growth. Literal meaning of cancer is uncontrolled growth, uncontrolled proliferation, uncontrolled multiplication. That is called as cancer. That is the literal meaning of cancer. Here this myeloma cell is one type of cancer cell. Now this myeloma cell can undergo multiplication, can form many cells. It has division capacity, it has multiplication capacity, proliferation capacity. Now in this hybridoma technology, what we are doing is, the B cell is merged with the myeloma cell. The B cell can be merged with the myeloma cell, thereby we can generate a hybridoma cell. This is called a hybrid cell or hybridoma cell. Hybridoma cell. So, by providing suitable conditions, the B cell can be merged with the myeloma cell, thereby we'll get a hybridoma cell. This is called hybrid cell. Now, this hybrid cell is having two characteristic features. One is antibody producing capacity, another one is multiplication capacity, proliferation capacity. So these two characters are individual. Okay. So the B cell is having antibody producing capacity, the myeloma cell or cancer cell is having multiplication capacity. Now, excuse me. Now in this hybridoma technology, we are merging this B cell with myeloma cell, thereby we are creating hybridoma cell. This is a hybrid cell. So in this hybrid cell, we are getting two characteristic features. One is antibody producing character, another one is multiplication capacity. Okay. Now this hybridoma cell can be generated. Further, this hybridoma cell is grown in the laboratory by providing suitable nutrient medium. Okay. So in the laboratory, suppose say this is the nutrient medium. So in this nutrient medium, the hybridoma cell can be grown. So if you provide the suitable nutrients, the hybridoma cell, as it is having multiplication capacity, so it undergoes proliferation, it undergoes multiplication. So thereby group of cells are formed. Many number of cells are formed. All these cells are similar in their structure and function. So this is called as clone. This is called as a clone. And now these hybridoma cells, they are also having antibody producing capacity. So they releases the antibodies into the medium. They releases the similar type of antibodies into the medium. And these antibodies will be collected and they get purified. These antibodies get collected and they get purified and they can be used for treatment of the disease and they can be used for diagnosis of various types of disease. Suppose, say for example, if you want to raise the antibodies against hepatitis B, simply you infect this B cell with hepatitis B virus. Okay, when you infect this B cell with hepatitis B virus, the B cell get activated, it releases the antibodies against hepatitis B. In a similar manner, suppose if you want to raise the antibodies against another particular type of disease, just to infect this B cell with that pathogen. Okay, you will get the antibodies against to that pathogen. Are you getting? So like that, we can produce n number of monoclonal antibodies against to a particular disease. And these type of antibodies which are produced, they are called as monoclonal antibodies. So they can be represented by MABs. MABs means monoclonal antibodies. Now these monoclonal antibodies played a crucial role in this pandemic situation. Okay, because these acts as immune boosters. These acts as immune boosters. Okay, monoclonal antibodies. So for example, various kinds of antibodies are available in the market. For example, trastuzumab. Trastuzumab. It indicates with the suffix MAB. Pertuzumab. So it indicates with suffix MAB. MAB represents monoclonal antibodies. So in exam point of view, just you have to remember what are monoclonal antibodies and how they are produced. Okay, what is the method involved in the production of monoclonal antibodies? That is simply hybridoma technology. So this hybridoma technology is used to produce monoclonal antibodies. Got it? Just I'll revise once. So what are monoclonal antibodies? Homogeneous preparation of antibody molecules from a single clone of B lymphocytes. Homogeneous preparation of monoclonal antibodies from a single cluster or single clone of B lymphocytes. That is called as monoclonal antibodies. Generally, in our immune system, we have WBC cells, that is lymphocytes. These lymphocytes will release the antibodies, particularly the B lymphocytes. And the antibodies released by these B lymphocytes are polyclonal in nature. Naturally producing antibodies are polyclonal in nature, but artificially we are producing the monoclonal antibodies by hybridoma technology. So these monoclonal antibodies can be produced in large scale against a particular pathogen, against a particular antigen. Okay, the process involved in this monoclonal antibody production is hybridoma technology. What we are doing in this hybridoma technology, simply we are isolating the B lymphocyte cell. And these B lymphocyte can be infected with a specific pathogen. Thereby, it get activated. And this B lymphocyte cell can be merged with the myeloma cell. Myeloma cell means cancer cell. Cancer cell means literal meaning of cancer is uncontrolled growth. Now, this myeloma cell is having the proliferation capacity, multiplication capacity. When you merge this B lymphocyte with this myeloma cell, you will get hybrid cell. This hybridoma cell is having two characteristic features. One is antibody producing character, another one is multiplication proliferation character. So when you grow these hybridoma cells in the laboratory by providing the suitable nutrient medium, So these hybridoma cells get multiplied. So many number of hybridoma cells are found. All these cells are similar in their structure and function. They release a similar type of antibodies. And these antibodies we call as a monoclonal antibodies. Monoclonal antibodies. So these monoclonal antibodies are used in the diagnosis of, they can be used as diagnostic kits to identify the disease. So they can also be used to treat various types of diseases. Okay, like hepatitis B. Hepatitis, this is a monoclonal antibodies. Right.